about chapter 6 today and talk about the visual system. I will incorporate some things about perception. And as a neuropsychologist, we do view these things as separate but related concepts. And more broadly, we talk about vision, we also talk about perception, we also talk about visual construction, and things like visual motor integration. And we think that these are all really important skills to understand, some of which can only really be understood by integrating multiple things, the visual system along with, say, the frontal lobes or executive functions like motor planning. But I'm going to talk a little bit about construction and, or I'm going to talk a little bit about perception, less about construction, focus on vision as a sensory phenomenon. So, vision, in terms of motivating its importance, vision subsumes almost 20% of the cortex. I mentioned that a huge amount of human cortex is devoted to association and higher order intramodal, multimodal kinds of cortices. But vision still does have a large cortical representation, especially if you look at the other lobes. If you look at, for instance, how much of the frontal lobe is primary motor area, or in a more significant case, how much of the temporal lobe is actually devoted to primary auditory cortex. Most of it's not, right? But vision has a lot of cortical structures associated with it. So it's obviously important for how we function. And we are visual creatures, and when you think about our sensory skills, our vision is our dominant sensory skill in many ways. We see a lot better than we hear or smell. If you think about animals, a lot of animals can hear and smell things that we can't hear and smell. They can understand fine-grained gradations, like for instance the way dogs can smell. They can do a lot more with their smell sensation than we can. But vision is something we can actually do quite well. Human vision is really well developed for some very specific reasons. And here are 3 examples. One major advantage to human vision is our ability to forage, and we'll talk more about this. So we need to be able to tell good food apart from bad food. And that gave us a competitive advantage that gave us an ability to maintain in the environment, a niche, right? Because the better we were at finding edible food, the more edible food went to us. And so we have a strong ethological benefit in foraging for edible foods and differentiating edible foods from unsafe foods. And so this in particular is going to also emphasize things that from an ethological or sociobiological standpoint, you'd expect women might be slightly better than men in this aspect of our visual skills. You expect men would be slightly better than women in this aspect of our visual skills, because distance acuity is also very important for our functioning as hunters. And again, we are adapted to engage in this long-range pursuit hunting of animals that humans really do better than any other kind of animal. So distance acuity for hunting is also an important reason why we have good vision. And then finally, there are the social reasons why we have good vision. A lot of advances in humanity compared to the great apes have to do with our more complex social structures, which allow us to pass on information, which allow us to team together to mutually benefit, to engage in things like kin selection. That is, when we work together, we allow other people who share our genetic code to reproduce, which confers an advantage, competing advantage for our genes. So things like facial recognition and body language monitoring skills allow us to do complex functional nonverbal communication, both to send messages nonverbally and to receive them, and to interact socially, right? So this allows us to engage in much more complex social rituals than, for instance, the great apes do. So we've got some really good reasons why we would have good vision. In terms of I'm going to talk a little bit about physics, and I'm happy to take some time to explain these concepts. This is a psychology class. I want to focus on psychology, but vision is a sense, and so you want to understand the environment that is being sensed. So whether or not it's through view, I do want to point out that light is a fundamental phenomenon, right? Light is composed of photons, and more particularly, light is composed of photons that form into transverse electromagnetic waves. And what's important to know about this is that all transverse electromagnetic radiation, in principle, is the same thing as light. What differentiates TEM waves is the energy, the photon energy. And just as a physics review or primer, the wavelength, the frequency, and the energy of a photon are all directly related. Long wavelengths are low energy and low frequency. High wavelengths are high wavelengths are short frequency. Long wavelengths are high frequencies and high energy. Short wavelength. This is not right. Okay, let's not say this right. And fix it right now. So. Great, that makes more sense. A low wavelength, red light, has a high, is this still not right? Okay, sorry about that. Let me make sure I say this right, and make sure this makes sense. So, I may need to turn this off for a second and draw a picture to make this clear. So this is a little bit of physics in a really hand-waving sense. So the reason you call them transverse electromagnetic waves is, in essence, in 3 dimensions, you have 2 components of the wave. So I'm not sure I'm going to be able to draw this in a way that works, but hopefully I can kind of make this look 3-dimensional, which itself is a visual trick, right? It's called a transverse electromagnetic wave because you have electricity and the magnetic field are perpendicular to each other. Okay, so that's not super important for us as psychologists, but that is important in the sense that, in principle, a gamma ray that has gamma rays that have a transverse electromagnetic structure, you slow them down enough, you take enough energy out of them, they're light. So the wavelength is the amount of space that one full cycle, I should draw it this way, one full cycle takes, right? If you remember your basic sine waves and cosine waves, right? So the reason it's called a wavelength is this is the whole thing, right? It just happens again here, right? And so all light travels in vacuum at the speed of light, 3 times 10 to the 8 meters per second, right? And so this distance is typically for visible light on the order of hundreds of nanometers. So like 500 nanometers is going to be right in the middle of the visible spectrum, right? So just to give you an idea of what 500 nanometers is like, 1,000 nanometers is 1 micron, or 1 micrometer. Human hair is about 100 microns thick. I think a page is about 100 microns thick also, a page of paper. So that's 100. You divide that by 100 in your head, that's a micron. This is in the infrared, essentially 1 micron wavelength. 500 nanometers, half of that is in the middle of the visible spectrum. So there are a couple hundred wavelengths of light in the thickness of a sheet of paper, on the order of hundreds of wavelengths. So this is a small quantity. Okay. Just to sidetrack briefly into physics, what's important about this also is that you can't represent stuff that's on a smaller size scale with this kind of light than this, because this light defines a space. That's a quantum mechanical concept. This light essentially, this photon has a width that's associated with its wavelength. So that is, for instance, why you use X-rays and stuff like that to do imaging, in part because they can transmit through things, but also in part because they're high frequency, so they have small wavelengths, so they're small, and so they can image in fine detail. So this is a wavelength. usually referred in physics as lambda. And you can imagine there's a bunch of these wavelengths here, right? So this stuff is moving at the speed of light, 3 times 10 to the 8 meters per second. So you imagine in a certain unit of time, you can imagine how many of these wavelengths are passing through a certain space, right? The longer the wavelength is, the fewer the wavelengths will pass through that unit of space in a given time. That's why a longer wavelength is associated with a lower frequency because less waves pass through the space. And it's a little bit more complicated to explain why a longer frequency is associated, or a lower frequency or a longer wavelength is also associated with a lower energy density. The photon in this field is at a lower energy level than a bluer photon, like at 300 nanometers. Don't worry too much about that. But let's double back here and make sure I get this right. Okay, I think I see what I did here. This is where it became confusing. I actually was changing the one thing. I only had one word wrong, not all three. Okay, so this should make sense. Let's check this and make sure this makes sense with the explanation I just gave you. A short wavelength is a high frequency because in a unit of time, lots of those wavelengths are going to pass through, right? Because they're coming at the same speed, and you get more wavelengths packed closer together. That high frequency is associated with high energy. So I should have said short instead of low wavelength. Short wavelength, high frequency, high energy. Long wavelength, low frequency, low energy. Short wavelengths are blue, or ultraviolet. Long wavelengths are red or infrared. So this is the range of light that humans can see, somewhere around 400 nanometers to somewhere around 750, 780, something like that. You may be able to visualize some stuff further out than this weakly, but most of what you're seeing is like this. And so for instance, just to sidetrack a little bit more, if you've ever heard of black body radiation, this wavelength range that we can see explains the colors that you see in hot objects. You can see light in a certain range from, and this is what I'm drawing here is the wavelength. So a short wavelength, this is blue and this is red, okay? And you can see between blue and red essentially, right? Any body has, any structure has a thermal energy profile, and that thermal energy profile has a kind of curve, something like this. That curve moves to, that curve is actually, for wavelengths, it's going to be over here somewhere. As that object gets hotter, there's more energy in the object. It releases photons that are at a higher energy, so it moves to shorter frequencies. Again, short frequency, high energy. Then that object enters the visible range. And this is long wavelengths. So objects that are a little hot are red hot, right? Later, that curve is essentially, it's big enough that it spans across the visual range. And if you remember, white light is light that's composed of all colors, which is what you've got here. So white hot objects are hotter than red hot objects. And very hot objects now are emitting black body radiation that's at such a high energy level that you can only see the low energy tail, and that tail is going to be at the blue end of the spectrum, the highest energy light that you can see. So blue hot is even hotter than white hot. And that's where that comes from. The reason there's red hot and white hot and blue hot has to do with what you're able to see and how that overlaps with the energy that's being released from a hot object. So, just for fun. So you can see light from these blue, purple, ultraviolet kind of wavelengths up to the red. And your vision is pretty high precision. Within the focal plane of your vision, you can readily perceive sub-millimeter scales. Remember I mentioned that human hair is in the hundreds of microns kind of range, like a couple hundred times the wavelength. So you can see at this kind of size scale. That's actually pretty amazing. When we talk about some of our other sensations, just stop and think about how fine-grained some of the sensations are. Like you can feel roughness and texture that's in the order probably of microns, which is pretty amazing, actually. So you can see very small structures. Like if you have a fancy phone, Apple had this big thing with a Retina iPhone like 2 years ago or something like that. And the whole idea was that the phone was designed so that when you hold it at a normal using distance, the pixel density was so high that you couldn't perceive it. You needed to have a ton of pixels. Like 5 years ago, you had a monitor that was 30 inches across that had as many pixels, or something like that, probably not 5 years ago, but not so long ago, like the monitor that was 30 inches across had as many pixels as an iPad did. And that's how much you had to shrink and increase the resolution to catch up with the human eye. And that's not about iPads, but this is about what the human eye can do. You had to pack a lot of pixels into that device to make them smaller than human eyes can see. And even at those kinds of pixel densities, you can't see the individual pixels, but you can still tell, you can perceive roughness that has to do with the resolution limitations of the device. So very high resolution. And actually also very sensitive to small light sources, weak light sources. And we probably are not able to see single photons very often, maybe in some limited cases, but the number of photons that we need to see within a 100 millisecond time window, probably in the order of 5 or 6. So 5 or 6 individual particles of light, or individual quanta of light. That's a very, very, very weak light source. And you can actually see that. So we actually have a really fairly remarkably advanced vision system. Like another example, just to think about like our dark adjusted vision, being able to see very weak light sources. You probably all own cameras of some kind, right? Like a cell phone camera at least, right? Have you ever taken a picture with your camera in the dark? Like at night? Yeah. Those pictures kind of stink, right? Like they usually don't come out very well. Think about how much better you can see in the dark than the best cameras that we can make. You can actually see really remarkably well in the dark. And you can see remarkably well in the light too. When it's too bright outside, the cameras don't do very well either. And we can handle all of those situations. So the visual system from a physics standpoint is really, really well designed, and particularly in terms of precision and sensitivity, or dynamic range. Sometimes people in photography will talk about. So a camera, it's hard to build a camera that can see very bright things and very dark things. The human eye is actually such a device. In perspective, so this is what I mentioned earlier, transverse electromagnetic radiation at any frequency is conceptually the same thing. It's just a difference of how much energy there is in the photon. Kind of like your neurons just fire or they don't, and they transmit the rate information. It's just this frequency information that really distinguishes different electromagnetic radiation. And so, like, we talk about blocking UV light, like you wear sunglasses that have UVA and UVB protection, you wear sunscreen. That is light. It's just light that's outside of your visual range. The frequency is too high. It's too high. The wavelength is too short. The energy density is too high. So you can't see those, but you can absorb them, and they cause problems. Part of the reason you can't see many of these is also because a lot of optical surfaces absorb them. Yes?
What biologically limits you from seeing wavelengths at 300? Is it just that your neurons don't fire?
Well, so part of it is that each neuron is designed to have a distribution of frequencies that it responds to, and that's based on chemistry. Then part of it also is that a lot of this stuff is actually absorbed by optical materials, like glass. The reason you don't need, there's a reason you don't need to wear sunscreen inside the house. Part of the reason is because the windows in your house will block UV light, will absorb it. So your eye will actually absorb some of that light also. That's part of the reason that you can't see some of this. But then the other part of the reason is that there's a chemical wavelength profile that each rod or cone can visualize. and it only breaks down for that light range? It's a really good question. So there are animals that can see some of this stuff, and there are animals that can see some of this stuff. Not so much out here somewhere, but in this kind of range. So some animals actually have a wider range of light that we can see. The other thing to know about light, just in terms of the physics, is as far as the hue of light is concerned, hue is made up entirely, from a physics standpoint, of the frequency distribution of light, which is this one variable, the wavelength. So any color of light in principle is some distribution of wavelengths across this range that we can see. Or if we were not a human, for another animal across a different range than we can see. There's actually just the one variable, which is the frequency. Humans, in contrast, perceive light based on three-dimensional properties. And the hue, saturation, and value, sometimes like you'll see HSV on your computer. That's one way of thinking about it. So hue has to do with a dominant wavelength of light. And that establishes the idea of color. But we can perceive lots of different colors. I didn't put that on the other slide, but we can probably perceive several million colors. Saturation has to do with the relative purity of the dominant wavelengths. So white light is defined as being highly unsaturated, or it has many different frequencies, whereas light that's purely one frequency is going to just have that one color. It's going to have a very simple color profile, right? So, and then value, which is brightness or energy density, how many photons there are. So if you've ever played around with colors on a computer or in a paint lab or something like that, if you adjust these three things, the hue, the saturation, and the value, you can achieve any visible color. And similarly, as we'll look at in a second, you can achieve any visible color by combining light that is red, green, and blue. And we'll see just in a second why that is. I do want to mention that if you have background in art, people in science will often say that red, green, and blue are the fundamental colors, meaning these are the colors of light that the human eye can perceive. There actually is no such thing as a fundamental color, but they're fundamental to us. In art, you'll talk about fundamental pigments, and the reason is because this is an additive logic. You're putting light together. In art, you have a subtractive logic. When you have a surface, you're interested in what light gets reflected off the surface and what light gets absorbed in the surface. And so you're interested in what light is not reflected and what light is reflected, which means that it's subtractive. The more different paints you add to the surface, for instance, the more light gets absorbed. That's why when you add paints of many different colors, you get black, but when you add lights of many different colors, you get white. Okay. So in art, people will talk about the three primary pigments, although sometimes they say colors, and those are cyan, magenta, and yellow. And cyan, magenta, and yellow essentially sit in between. Like if you draw a color wheel, there's red, green, and blue, and then cyan, magenta, and yellow are just in between the colors. So cyan is kind of a light blue, magenta is kind of a purpley pink, and yellow is yellow. But we're talking, when we say fundamental colors, again, that's human talk. Light is a frequency distribution across these wavelengths, but humans have three cones that see different colors of light, and so you can break light down into the responsiveness of each of those cones. And as long as you match that responsiveness profile, two different wavelength distributions will look the same to us. We can't tell the difference between them. So here is that pictorially. Hue, saturation, and value. So the value is represented here because these colors are all shades of this. What's happening is here you're decreasing saturation. So this color is very white because you're adding lots of different wavelengths to it. Here you're decreasing brightness and increasing brightness. And so here's the highly saturated color with just the one kind of color. Yeah. So you take away the light, it gets dark. You add the light, it gets white. And again, you could also do the same thing by adjusting red, green, and blue in terms of light. You can achieve the same thing on a surface by adjusting cyan, magenta, and yellow pigments. Does that all make sense? There's a lot of physics and art and stuff like that. But all of this is driven, what is very important is all of this is driven by how we see. Like the idea of fundamental colors, again, comes back to how humans see. If we saw in a different way, we wouldn't have this concept of fundamental colors. And I think, if I remember correctly, there are actually some birds, like birds of prey, that have more than 3 kinds of cones. And so essentially they have 4 or 5 fundamental colors because their color distribution is broken out into the sensitivity of 4 or 5 cones instead of 3. And then some animals have 1 or 2 cones or something like that. The visual hardware. Some important things about eye construction. The eye functions as an optical device all the way through to this rear surface. What I mean by that is it's all physics in here. This is a lens. It operates according to the same physical properties as a glass lens, like in your camera. This iris functions much in the same way that the shutter in your camera functions. This is a fluid-filled surface, a fluid-filled body, but here light is coming in and it's focused back on the retina, and it's actually focused in the sense of a picture. And if you look at old experiments, you cut an eye open, you can put like sort of imagine a movie screen being put here. You actually are seeing a picture that's focused on this plane that you're seeing. So this is just a visual picture that's physically being transferred here, up until the retina. The retina is where the transition happens from optics to the electrochemical neural circuitry. So the rear surface of the brain, in the eye, is organized inside out. This is an important concept. The rods and cones are actually on the rear surface of the eye, and a lot of this other stuff is actually in front, just kind of a strange organization. But so light is passing through ganglia cells and stuff like that to get to the rods and the cones. And I'll show you a picture of that in a moment. And those rods and cones then are back here against the rear surface of the eye in the retina. And that's where the picture is being transferred. And then through these axons are projecting into the optic nerve, it's going back into the brain. The retina is organized with a central fovea. The fovea has a high density of receptors for high acuity objects in the center of the visual field. I think there was forget exactly, like there's a grape at a kind of arm's length-ish distance. That's how big your foveal vision region is. It's very small. And that region has a disproportionate emphasis in our visual processing. We see very, very well. And when you think about your perception of the room, there's all kinds of stuff going on in the room, right? Most of it's bigger than a grape held at arm's length. So what you're doing very, very well is moving your eyes around and adjusting the position of that fovea and integrating information so that you can see a wider space and high resolution. So central fovea, and then blood vessels and axons collect out of the eye at the optic disc, which results in a blind spot. And if you do the right kind of trick, you can actually visualize that blind spot where you can't see. But most of the time, the other eye is compensating for it, so you never notice that you have a blind spot. So let me talk briefly about oculomotor control, so how we move the eye around. As I mentioned, that's really important in the way we see. Let me mention that there are 3 pairs of muscles that are manipulated by the cranial nerves. And so this muscle is essentially, I don't have a picture of this in my lecture, but those muscles rotate the eye like this. So I look to the inside and to the outside. They rotate the eye essentially kind of in up and down. And then for complicated reasons, you have a 3rd set of muscles that rotate the eye at an angle. And those are the oblique muscles. So 3 sets of muscles that allow the eye to position the retina so that you can see objects in the foveal region in a wide variety of space, and you have peripheral vision also. If you've looked at comparisons of humans and animals, we also just briefly mentioned that predatory animals have a different eye structure than herbivores generally, right? So like if you look at a rabbit, the rabbit has eyes that are more on the side of its head. The rabbit has a huge peripheral vision, like they can see back here. Humans and dogs and other creatures that eat meat have eyes that are generally pointing forward in the front of the head. So the vision of herbivores is optimized to detect prey, to detect predation at different angles. We're optimized more to be seeking out something so we can decide where we look. And so we're better at this kind of focal foveal vision. And we move our eyes around where we need to. We have muscles for opening and closing the eyelid. The levator palpebrae is the muscle that opens the eyelid. There's some diseases that we won't get into in this class most likely that involve impairment of these muscles as a characteristic feature. You've got muscular control for adjusting the lens curvature. And so there's a set of radial muscles that pull the lens to make it flat or release the lens to make it more bubbled. So remember, a lens that has a high radius of curvature has a short focal length. A lens that's almost flat has a very long focal length. So adjustment of the lens curvature will change the focal plane. So changing focus from far away to close up. And that's called monocular accommodation. It's called monocular accommodation because just one eye is doing the accommodation. There's also binocular accommodation. So for instance, if I am looking at an object here, I need my eyes to be oriented such that my two foveal regions are looking there, right? So they need to be cross-eyed a little bit. And the closer I get, the more cross-eyed they need to be. But the further away I get, they don't need to be so cross-eyed. And at infinity, they wouldn't be, the lines don't diverge at all, right? They're parallel, and so the eyes would also be parallel in their orientation. That's binocular accommodation. So you have different techniques for adjusting the eyes to be able to see at distances. And then finally we have muscular control to widen and narrow the iris to adapt for dark, bright situations and manage depth of field. If you have ever played with a sophisticated camera like an SLR camera, you may know a little bit about how this works, but suffice it to say that this iris is a ring, basically, that controls how big the opening is. And when the opening is very small, you don't let very much light in. For complicated reasons, that also means that you have a wide depth of field. You can see things that are farther and nearer away, and they'll both be in focus. When you open it up, you let lots of light in. You have a very narrow depth of field, and so you can do two different things with this. But so, like your doctor does the fundoscopic examination, they shine the light in your eye, right? And they shine the light to see if your eye constricts. That pupillary response is a change of the iris. That's what's constricting. Oculomotor control is directed. All this stuff is controlled at the brainstem level by cranial nerve nuclei and some other nuclei in the brainstem. And so the brainstem can do a number of things. One thing it can do, for instance, is handle visual orientation in real space as the head rotates. And so you can actually have a person who has a large portion of their brain inactive due to a serious head injury. And if you turn their head, their eyes will stay pointing in the same direction. And then there's also brainstem control to guide the eyes to a change in the visual field, like something moving. And both of these things actually are inhibited by higher cortical structures, because you don't want to look every time something happens, right? You should be able to not look when something happens. It's the frontal lobe that inhibits these things. And then the frontal lobe, then in general, kind of consequently, is responsible mostly for intentional placement of focus. So smooth pursuit, directed vision, that's the frontal lobe. It inhibits these more automated responses. And so eye motion is a mixture of jerky motions and smooth motions. The jerky motions are called saccades, and you do them constantly. You do them without thinking about them. And this again is what allows you to sample different parts of space to allow you to have a high resolution representation of something larger than your phobia. And also to detect whether there's motion happening in that space, whether there's something that you should guide your attention to. So saccades, for instance, are really important in reading, because as you learn to read, your saccades will follow a different pattern. Like most of you are good readers, you can read at a sentence level or even a paragraph level. And so as you're reading a word, but you're looking ahead to see what that word is in context, right? Like a word could mean multiple things, like ring could mean a ring you wear on your finger, or the ring of the iris, or it could mean ringing a doorbell. So you're reading ahead to find context to see what ring they're talking about, right? That's a saccadic activity. So your visual saccades are being used to facilitate your reading ability. And then smooth pursuit, of course, you go where you want to. So that's the way we control our eyes. I want to mention, see if this video works. There are some neurological disorders. If you get to understanding basic neurological disorders, you'll talk about the cranial nerves a lot because they do a ton for diagnosing. And so there are a bunch of visual effects that have to do with the way we move our eyes that can be really useful in terms of identifying brainstem abnormalities as well as frontal lobe abnormalities, because again, these two structures work in conjunction. And if this works, I want to show you an example. This is a test for something called optokinetic nystagmus. And essentially what this has to do with is as you're following something in space, like a train going by you, like that train goes by, and you can sort of imagine like the cartoon character who watches the train and then their head's stuck over here, right? But the train keeps going by. So what your eyes actually do is they beat. They follow the train and they keep following the train by beating back and following the train, beating back and following the train. So this is called optokinetic nystagmus. Let's see if I can show this to you.
Test the same system by using an optokinetic tape, which has various shapes. And what I want you to do is I want you to look at each new shape as it comes and appears. So looking right here, keep watching it move it
well. Oh, there you go.
to the right.
See how our eyes are beating?
I'm going to move it to your left. Keep watching right here.
See the beating back and forth?
Very good. I'm going to move it up. Again, watching the new object as it appears. And we're going to move it down. Again, watch the new object. Very good.
So the most obvious kind of pathologies that you would be looking for in that kind of case is both of those eyes should be doing that in concert because they're seeing the same thing and they're seeing it together. If one eye beats differently than the other eye, that's a sign of a neurological abnormality. But so this is actually useful. Doctors use this. Yeah.
So people who are dyslexic, does that have anything to do with this control of the eyes, or is that something completely?
You know, there actually is research that looks at saccadic eye movement and reading disabilities like dyslexia, and there is some evidence that there are abnormalities in the way that information is processed. Yeah. So your eye movements change over the course of your reading skill development. Like we talked about this concept elsewhere. Like I think I mentioned in the first lecture that the way you process music activates different cortical areas if you're skilled as a musician than it does if you're naive to music. In that same way, as you learn to read, your saccadic structure actually modifies, and it optimizes the reading process. And you have reading-specific saccadic motions. And then in dyslexia, you don't see the development of those. Yeah, that doesn't mean dyslexia is an eye problem, but there is a coordination of the sense with a higher level perceptual activity that's involved in reading. And actually, just as a side note on that, there are some people who say that there are kids who are misdiagnosed with ADHD also, because actually what their problem is convergence. And so they're having difficulty focusing in different visual depths. And so the idea is that this visual deficit causes attention problems. And if you in this case, if you fix the visual deficit, you fix the attention problems. And the problem with that is that the people who are testing your eyes are usually not testing your auditory attention or another kind of attention, as they haven't really proven that it's an eye thing. But there may be some kids like that. But so in the case of reading, we don't know, where that comes from. Like I think the question would be, are there saccadic abnormalities outside of reading? If there are saccadic abnormalities outside of reading, then the argument would be that something is going on that doesn't fundamentally have to do with reading, and maybe there could be like an ocular therapy that could help correct that. So here's a picture of that retinal surface, and again, the back of the eye is where the photoreceptors are, and then there are these interneurons. Remember that word? So bipolar cells connecting from neuron to neuron. This is a local interneuron because it's connecting between two neurons that are in the same structure, right? And similarly, this horizontal cell. So the bipolar cell is connecting the ganglion with the photoreceptor. The horizontal cell is connecting the photoreceptors together. The amacrine cell is more connecting the ganglion cells together. So these ones in the middle, the blue and green and red, they're all interneurons. And I guess technically the ganglion cells are interneurons also because they connect these cells with the central nervous system. So these guys, if these guys are all local interneurons, these guys are essentially relay interneurons because they're going from the eye somewhere else. So as you probably do know, there are there are two categories of photoreceptors in the eye. They're cones and rods. And cones are for seeing color, and rods are for seeing in black and white, or monochromatic. And we'll talk more about the specifics. There are different kinds of cones to see different kinds of colors. That's where the trichromatic color theory comes from. That's why we think of color in three fundamental colors, because there are three cones. And these cells are aggregated, they're integrated, that information is ultimately sent in for perception. There is some perception of features that happens at this level, but most of that is happening in the brain. So, cones and rods. These terms, I don't know that if these two terms are in the book, but they're important terms to know. Photopic and scotopic. Photopic means light adjusted, and scotopic means dark adjusted. So, cones are photopic, meaning they are good at seeing in bright situations, and rods are scotopic, meaning they're good at seeing in dark situations. So that those are important terms to be aware of with the vision system. So these are the two photoreceptors, and again, there are three different kinds of cones that essentially see red and a green-yellow kind of color and blue. There are ganglions, there are interneurons, bipolar cells, ganglion cells, horizontal cells, amacrine cells, and the bipolar cells connect the photoreceptors with the ganglia. The ganglia cells source fibers for the optic nerve, so the optic nerve is made up of the axons of ganglions. And remember, that makes sense. Information goes down the axon, down the cell body, right? It's going from the eye to the brain. And so the source of the fibers in the optic nerve is the ganglion cell in the eye. The destination of the optic nerve is the lateral geniculate nucleus and so forth inside the brain. And so there are different kinds of ganglia. Some ganglion cells aggregate information. So for instance, the rods are very sensitive to low levels of light, but in order to be able to see at those very low levels, you want to aggregate across them or integrate so that you're using multiple detectors. They can all see a very small amount of light. You're pooling the signal to increase the signal level. And then also to process information to a very limited extent. In the foveal region, the ratio of cones to ganglion cells is more like 1 to 1. And so the ganglion cells are integrating information across cones, not for the purpose of amplifying it, but for the purpose of doing some pre-processing, essentially. Horizontal cells connect across photoreceptors and or across bipolar cells. Amacrine cells connect across bipolar cells and ganglion cells. Again, horizontal, amacrine, they're connecting cells locally, so they're local interneurons. The three photoreceptors have different peak densities, and this gets to your question about why we can't see colors outside of this range. So each of these photoreceptors has a chemical photopigment, rhodopsin, which is made of this opsin and the retinol. The opsin is different in each receptor, chemically different, and the retinol is the same. And so each opsin has a likelihood, like our cross-section, a likelihood of breaking down the rhodopsin into the retinol and the opsin that varies with wavelength. So at this peak wavelength, the likelihood of absorbing the light and breaking down is the highest for the blue cone, and at this peak for the green cone, and at this peak for the red cone, and at this peak for the rod. These numbers are slightly different from the book. Don't worry too much about that. These are generally the locations of these around 420, around 500, in the 520, 530 kind of region. 560 kind of region. And they are wide distributions, right? So that's important to note. Like, look at the distribution for the red cone, right? And the blue cone. There's light. Like, light right here is stimulating all three of the cones. And so green light will stimulate the red cone, but just not as well as it stimulates the green cone, because the green light is here somewhere. The likelihood is higher for the green cone than for the red cone. It'll stimulate, some light will stimulate the blue cone, even though it's more green than blue, but it'll stimulate the green cone better than the blue cone and vice versa, and so on. And so this is how you see color. We talked about how color is actually a distribution across all these wavelengths. It gets resolved into how much it makes these three cones fire. And so the relative ratio of firing of the three cones is how we see color. Excuse me. So again, the light decomposes the rod opsin into the opsin and the retinal, and that modifies the photoreceptor's firing rate. I do want to mention again, I mentioned this briefly, I think, at the beginning of class. This is kind of a unique situation in the nervous system. So the rod opsin is kind of like a conductive switch. When it's together, it allows stuff to go through. So when the rod opsin is intact, the neuron actually fires more. When the rod opsin is broken apart, the neuron actually fires less. Just like if you unplug something, you unplug it, the stuff stops working, right? My laptop shuts down or whatever. So photoreceptors, unlike a lot of cells, they fire at a low rate when there's no light, because then there's no, I'm sorry, let me say that again. They fire at a high rate when there's no light, because when there's no light, the rod opsin is intact. When the rod opsin enters the system, it breaks the light enters the system, it breaks the rod opsin apart. That decreases the firing rate. So it works backwards from a lot of other cells. The intact rod opsin results from the absence of light, which generates firing. And so when there's light, the firing slows down instead of speeding up. And that's related to, like, for instance, that tells you something about what the rods are doing during high light situations, like right now. There's lots of light in this room. Essentially, all the rod opsin in the rods is broken down. So the rods are firing at a very low rate. And this might be one reason why this is good, because otherwise all day your rods would be firing like crazy, because they would be seeing so much light. But instead, your system is built backwards so that your rods aren't firing at all during the day. They don't fire until the night when they're not saturated, because the rods are heavily saturated during the day. They're very light sensitive. There's tons of light in the room right now, right? There's a conceptual relationship. I mentioned that I would weave in these principles we've been talking about into these lectures to the all or none principle and to Muller's doctrine of specific nerve energies. And what that has to do with is that photoreceptors only fire or do not fire with a rate dependent on the incident photon density and wavelength. What that means is that red cones, when they fire, tell your brain that you're seeing red light, even if they fire because you're seeing green light. That's all they can do, is generate a red cone signal. There's no way for the red cone to say, hey, I'm firing, but this is green light I'm seeing, right? So they either fire or they don't fire. It's the ratio of the firing that allows you to perceive color, but when the red cone fires, it transmits red cone information. It can't transmit green cone information any more than it can transmit sound. And that's a corollary, essentially, to the all or none principle, or to the doctrine of specific nerve energies. Okay, so photoreceptor activity. Photoreceptors fire more frequently when the rod opsin is intact, just like I just said. So incident light decreases photoreceptor activity instead of increasing it. So here's just a summary of a few things I just said. Rods are scotopic. They work in dark light situations. During bright light, they're saturated, so they're not firing. And then cones are photopic. They need a lot of stimulation, and so it takes a lot of light, so they're largely intact at night. And so they're firing at a higher rate, and then they decrease firing during the day. And then, so it's important to remember also that during bright light situations, the rods are saturated, they're not participating. During dark light simulation situations, the cones don't receive enough photons, they don't alter their firing very much, so they're not participating. So essentially, in very dark situations, you're seeing with rods. In very light situations, you're seeing with cones. Here's some information about what the distribution of those different rods and cones looks like. The important thing about this, I think fairly really important idea is these little dots are rods and these dashes are cones. What I want you to notice is where the rods and cones are. The rods are all in the periphery of vision. This is the fovea. There aren't very many rods in the fovea. The cones are mostly in the fovea. There aren't very many cones in the periphery. I don't know if you've ever tried this, but if you try to see something in very, very dim light, sometimes you can see it off axis. Like if you're trying to see it, you look over here and you look at it and you try to see it in your peripheral vision. Your peripheral vision in dark situations is more sensitive than your foveal vision in the sense that the rods are all in your periphery, and so you're not really using your fovea in that situation. So, and then your color perception is going to be the best in your center of your vision. So that's the important idea here. Rods in the periphery, cones in the fovea. And here's a picture again of that blind spot. So there's the fovea and the retina. There's this blind spot where the optic disc is, where the optic nerve is exiting. So you can't see there because there are no photoreceptors there, just these exiting axons and blood vessels. So the fovea is full of cones, the periphery is full of rods. The other interesting thing that the book does mention is that the three colors of cones are actually not balanced. So only 8% of your cones are blue cones, but your brain adapts to that. And you can see stuff in blue. You seem to see blue as an equal footing color with red and green. It's not like blue is really dim and red and green are really bright. In certain situations, for really blue things, you can actually, I think, you can actually see the effect of this. If you ever looked at really blue-purple flowers that have only these really high wavelength, or like a good example is little flowers that are these really ultraviolet-y blue, like cobalty color. If you ever try to look at them, they don't seem very sharp, like they look kind of ghosted, and that's because your cone density for blue is not very, I think that's because your cone density for blue is not as high as for the other colors. And so your visual acuity in principle is not as high for very blue things as it is for red and green. But in practice, for most things, there's a mixture of colors, and you see the three colors in equal footing. There are three colors. There are some specific color vision deficits, color blindness, that are associated with these. And one that I didn't mention, and I don't think the book mentions, but is interesting, is so men are often said to lack style, right? And sometimes part of like the reason that men may not dress, some men may not dress in a way that's considered color fashionable, is because of color blindness, because they're not seeing the colors in the same way. I've heard that one more wrinkle on this is that there are actually two variants of the green cone, and they have the difference in frequency is like 15 nanometers. I don't know if this is, I'm not sure about this one, but I've heard that because there are these two variants of green cones, essentially the people who have the two variants, which are mostly men, I think women pretty much all have the same variant maybe, but the two variants of green cones shift the green peak frequency. What that means is that the color balance for those people is different than for everyone else. And so that's going to change their sense of fashion, for instance, because they're going to perceive complementary colors just slightly differently. Anyways, three major color blindnesses. You probably are aware that color blindness is more common in men. These two are sex-linked. They are X chromosome diseases. Because women have two X chromosomes, it's really unlikely that they would both be defective. And so women don't usually have these diseases. They'd have to carry two bad copies. but men only have one X chromosome. So if you've got the bad copy of the X chromosome, you're out of luck, and you'll have these. Tritanopia is not on the X chromosome, and so it's actually equally distributed in men and women. So protanopia and deuteranopia, essentially the first color blindness and the second color blindness. In protanopia, the red cones have green opsin in them. And so, again, the cone can only fire the signal that it fires. And so what's going on here is you've got the green chemical, the chemical that's optimized for the green distribution in the red cones. That means that both your red and green cones are firing for green images. Deuteranopia, your green cones are filled with a red chemical, so both your green and red cones are firing for the red color. And then tritanopia is a faultiness or absence of blue cones. I've got some pictures. So here's a 2 and a 6, and so here again, the red cones are filled with green opsin and the green cones are filled with red opsin. So let's see if we can work this out. In protanopia, you're seeing green, you're seeing red and green as green. So you're seeing red and green as green, red and green. That should mean that the 2 becomes invisible. Let me see, maybe I have this backwards. I think this picture doesn't look, I think this is the red one. This picture doesn't look right on the screen. So red and green both show up as green. So in protanopia, you can see the 6, but you can't see the 2. No, I said that. I said that right the first time. Yeah. So this is the red one. It looks green, and so you can't see this in protanopia. And then in deuteranopia, you can't see this one. You can see this one. And here's another example of that. And so I want to mention briefly, if you want to play around with this just to see what these color blindnesses look like, there's actually this program called Color Oracle, and you can download it for Windows, Mac, and Linux. And what it does is it actually simulate what it's like to not see in 3 colors. And so you can see what it's like to be colorblind. There's some other interesting things. I saw somewhere a website that tests how well you can differentiate different colors. And so it tests basically your ability to perceive different differences in frequency. But yeah. It is slightly confusing, and I'm not colorblind, so I don't know exactly what it looks like. But the idea is that the red and green distinction is the problem for both protanopia and deuteranopia. It's just that because you have either green opsin or red opsin, but not both, you have relatively more sensitivity to certain parts of the wavelength spectrum where the red or the green opsin works, and so those other colors appear darker or less visible. And then this tritanopia is not very common. Yes?
That's kind of a weird question then. Does that mean the focus is the same? Like they still see, like they're red-green colorblind, that maybe one was off focus a little bit more than the other, or is it pretty clear? Like, do we know that at all?
So in your phobia, you have lots of red and green photoreceptors, and you have some blue ones also.
Right.
You don't have very many cones. They're all in the same place, they're co-located. So the focal plane is the same essentially for all the colors. There are actually, this is getting kind of overly complicated, but there is, colors do focus at slightly different focal distances for a given lens, but that effect is really, really small.
Okay.
So it's not going to really contribute very much. So basically, all of it is in focus, but what's happening is the light should be differentially triggering red and green cones, but instead that light is the same to them because they have the same sensitivity.
Okay.
That's what's happening. It's just kind of complicated. It might be worth playing around with something like this or that website I showed you, just to get an idea of it. And it's hard to keep track, and it's not, a super crucial point in this class, but it might be a good way to keep track of what the cones are doing and what the actual impact of that is, like how the physics relates to the the perception, the cognitive process. Okay, let's continue. So I want to start talking now about perception, essentially. And so now we're switching from cells that are primarily focused on just detecting that the light is present to cells that are doing something with the relationships of the light. And those relationships are what ultimately going to give us things like the perception of motion, the perception of forms or structures. The ganglion cells are the beginning of that. So if you look at old psychology, there was actually an argument over whether light was a trichromatic process or an opponent process. And the best example of how the opponent process works is the after image effect. So if you look at something that is really, really one color and you look at it for a long time, you take your eyes away, you see an after image in a complementary color, right? So that's where the opponent process idea came from. The idea that maybe what you see is not 3 colors, but you see pairs of opposing colors. Turns out they're kind of both right. The photoreceptors are trichromatic. The ganglion cells are opponent process cells. So the ganglion cells have these sender-surround systems where the sender and the surround are 2 complementary colors. And they more or less work on red, green, and blue, but you need one more color for complement, and that turns out to be yellow. Because when you look at the color wheel, blue's complement is in between red and green, it's yellow. Technically, green's complement is in between red and blue, and red's complement is between green and blue. But, so they work on pairs of complementary colors, and the way they work is that the cells fire for a certain color in the sender, and the opposite color in the background, or vice versa. Sometimes the cells will fire for what's in the background and not what's in the sender. And so these are generating features in a very basic level. Like they're detecting gradients because the color changes from here to here. What they're detecting is that this is not the same color, right? So it's the beginning of some kind of feature in the visual field. There are different kinds of these opponent process ganglion cells. There are on and off cells. So the on cell is looking for the presence of light here. The off cell is looking for the absence of light in the sender. And so there are different kinds of responses. So the on cell, when you shine the light in the center of the receptor field, it'll increase in firing, and when you take it away, you'll have a rebound effect and it'll have slow firing. When you shine the light in the surround field, the firing will slow down. When you take it away, there'll be a rebound of fast firing, and then it'll go back to normal. The off cell does essentially the opposite. You shine the light in the sender, and it slows down, and then has a rebound that speeds up, so like this. If you shine the light in the surround field for the off, then it does this. So the 2 important things are to note are that 1, you can detect the presence or the absence of the target color in the sender, and that 2, these cells respond both to that thing, like the presence of the light in the target area, and to a change away from that thing, right? So here, changing away from having light in the center field creates an abnormal pattern. See, because the firing rate here is not like it is before, for a little while. So, continuing, the optic pathway. So the optic nerves travel along the underside of the brain to the optic chiasm that's here. At the optic chiasm, part of the fibers decusate, meaning they cross the midline, and then the other part continues on the same side. And I explained that before, but the concept again is that when you're looking at right space, you're looking at right space with the outside of one eye and the inside of the other eye. Those 2 sides look over here. The outside of one eye and the inside of the other eye looks over here. And so you need to cross those over so that post the optic chiasm, you've got a representation of a visual field, like your left visual field or your right visual field, not of your left eye or your right eye. So that happens at the chiasm, and then behind the chiasm, the gross representation is of the visual field, but at the lateral geniculate nucleus of the thalamus, the LGN, there actually are still separations you have layers that are left eye, right eye layers. But the left eye, right eye layers are aligned so that a point in visual field space is represented in pieces that are adjacent to each other. So in this stack, this piece is looking over there, this piece underneath that is looking over there with the other eye in the same place. So at the LGN, the lower visual fields pass through the parietal lobe. Again, so frontal lobe, the temporal lobe is on the undersides, the parietal lobe is up here, and the occipital lobe. The lower visual fields pass through the parietal lobe on the upper part and come to the upper bank of the Calcarine fissure. This makes sense because the parietal lobe's on the top, and we said that the lower visual field